Thank you very much. It's an honor to be a part of ILAR's 60th anniversary, so appreciate that very much. And also appreciate ILAR's contribution as a collaborating center for the OIE, in which I have the pleasure of representing the United States at the OIE every year in May. And appreciate the contribution of a lot of collaborating centers, and especially ILAR's. Animal research is the foundation for many of the medical breakthroughs over the past century. Almost every present-day protocol for the prevention, treatment, cure, and control of human and animal disease, pain, suffering is based on knowledge attained through research with laboratory animals. To date, there's no comprehensive alternative for animal models in research. Certainly, computer models and cell cultures, as well as other adjunct research methods, are excellent avenues for reducing the number of animals used, but the pathway to fully duplicating a whole living system does not yet exist. As a veterinarian, I can say we do not want to use more animals than necessary in animal experimentation, and we want to limit as much as possible the pain and suffering of those animals. All animal research is subject to strict federal regulations. My own agency, the Animal and Plant Health Inspection Service, implements the Animal Welfare Act under one of the units which we refer to as animal care. Our regulations, which are considered more extensive than those covering human research subjects, govern the care and use of animals in biomedical research. Further, APHIS participates in international efforts on this subject, such as International Coordinating Committee on the Validation of Alternative Methods. You had asked me to speak on animal research trends that are underway. The guiding principles generally agreed upon, which we refer to as the 3 R's, which everybody has already heard here several times. The 3 R's are reduction, minimizes the number of animals used; refinement, employs techniques that reduce pain and distress; and replacement substitutes animals with non-animal methods or lower organisms. Not only does my agency develop and enforce regulations along those principles, but our science centers strive to uphold them in our missions of animal disease research, diagnosis, vaccine evaluation, and training. This work is done under the oversight of the institutional animal care and the use committees at the respective centers. We experience unusual challenges and opportunities in applying the 3 R's to traditional laboratory animals, as well as numerous species of farmed animals, including many aquaculture species and wildlife. For example, in the area of reduction, literature searches are conducted to reduce duplication of experiments. Statisticians are employed to design experiments with the appropriate number of animals. Pilot studies are encouraged to ensure that methods intended are valid for the larger study. Animal reuse or sharing is encouraged, and techniques such as use of plasticized tissues are used in training. In the area of refinement, we continue to validate the use of anesthesia and analgesia where appropriate in disease process studies. Humane endpoints are emphasized. Death or severe disease endpoints have been replaced in multiple assays. In addition, environmental enrichment is expected. In the area of replacement, for years we have used relative replacement methods such as cell culture isolation and monoclonal antibody production. An exciting area of absolute animal replacement for APHIS is the use of in vitro assays for animal vaccine batch quality control rather than animal vaccination challenge assays. We have developed and codified such assays, sponsored international conferences between regulators and vaccine manufacturers on their use, and published on the topic in peer-reviewed journals. Recently, APHIS has observed increased scrutiny of animal use in experiments. In some cases, public or interest group concerns have halted certain types of animal use. Animal use standards, both regulatory and voluntary, have also become more exacting. This is due to ethical, economic, and scientific considerations and increased information from animal behavioral studies. The scientific and policy communities should continue to emphasize the One Health approach and collaborate on refinement studies. They should publicize the animal experimentation as necessary, but also emphasize the importance of the 3 R's. It's important to stress animal research involves following strict federal regulations and adhering to voluntary guidelines from various laboratory organizations, such as the Association for Assessment and Accreditation of Laboratory Animal Care International. ILAR can continue its good work by facilitating communication among laboratory animal researchers that results in high standards of animal care. Again, thank you for asking me to participate in today's celebration. Thank you.